Okay, continuing with skeletal muscle. So I've already mentioned this in the previous video. The muscle will contract as soon as the troponin pulls the tropomyosin out of the way and the actin and myosin can form a cross bridge. It's automatic. So what's going to cause that troponin to pull the tropomyosin away from the binding site, exposing the binding site? It starts back at the synapse where the motor neuron interacts with the sarcolemma. For skeletal muscle, it's acetylcholine. Acetylcholine is the neurotransmitter that's released by the motor neuron when that motor neuron fires. It's produced by the motor neuron, stored at the distal end of it, and it's released to stimulate the muscle fiber. So what's going to happen is there's something called the resting membrane potential. Sodium, which is found everywhere, is not found inside of the cytoplasm, the sarcoplasm of muscle cells very readily. And that's because sodium-potassium pumps use ATP and active transport, primary active transport, and move sodium out of the cell on a regular basis. Sodium is a positively charged ion, and because it's a positively charged ion, that means that it accumulates outside of the cell, and the outside of the cell becomes positively charged in relation to the inside of the cell. The inside of the cell is less positive, a negative. This creates a potential difference. The membrane is not permeable to sodium, and so potential energy can be stored by this thing. It's called capacitance. So to let sodium get into the cell in skeletal muscle, it happens in one of two ways. The first way is a sodium channel opens in response to acetylcholine, and it facilitates the diffusion of sodium into the cell. When sodium is outside of the cell, the outside is different than the inside, and we call the membrane polarized. If we allow sodium to enter the cell by opening a sodium channel, it depolarizes. The outside becomes less positive, the inside becomes more positive, and now both sides are closer to the same. We call the sodium channels that open in response to the neurotransmitter ligand-gated channels or chemically gated channels. Now, the movement of the sodium into the cell and the depolarization of the inside of the cell causes electrically gated channels to open, or voltage gated channels. These voltage gated channels open to sodium, and sodium then floods in, and that part of the membrane becomes depolarized, which then opens the next voltage gated channels, which depolarizes that, etc., etc., etc. These channels close up very quickly. So what we end up with is a wave of depolarization moving down the cell membrane, down the sarcolemma, and down the T-tubules. Because as you remember, the T-tubules are, the lumen of them is outside. So the sudden influx of sodium moving as a wave of depolarization is called an action potential. When the action potential gets to the sarcoplasmic reticulum at the triad, at the T-tubule, it causes voltage-gated calcium channels to open, and calcium leaves the sarcoplasmic reticulum and joins onto troponin. When it joins onto the troponin pulls the tropomyosin out of the way. When the tropomyosin is pulled out of the way, the myosin and actin form a cross bridge, and we end up with contraction. So allowing a little bit of acetylcholine to open up some ligand-gated channels at the motor end plate causes a wave of depolarization and action potential and voltage-gated channels to open all over the cell, which causes calcium released at each and every sarcomere in each and every myofibril, which then causes exposing of binding sites and muscle contraction. It's like a chain reaction. This contraction will continue as long as the nerve impulse, right, keeps on releasing acetylcholine and keeps those channels open. If the nerve impulse stops, then the acetylcholine gets removed from the cleft by an enzyme called acetylcholine esterase. It decomposes the acetylcholine. Those ligand-gated channels close. The sodium is pumped out of the cell repolarizes. When the cell repolarizes, calcium then the calcium channels close. Calcium pump the calcium back into the sarcoplasmic reticulum. It leaves the troponin, which allows the tropomyosin to cover up the binding sites again. Cross-bridging stops, and the muscle relaxes. So this is kind of the order of things. The impulse gets down the motor neuron. The motor neuron releases acetylcholine. Acetylcholine crosses the cleft and binds to the receptors. The sarcolemma is stimulated, an action potential travels over the surface, a wave of depolarization, and into the transverse tubules. When that impulse reaches the sarcoplasmic reticulum, calcium channels open, calcium ions diffuse, facilitated diffusion from the sarcoplasmic reticulum into the cytoplasm that binds to the troponin. The tropomyosin get pulled off the binding site, exposing the binding site. Cross bridges form, and then contraction occurs. Acetylcholine esterase gets rid of the acetylcholine. The calcium ions are transported back to the sarcoplasmic reticulum. ATP breaks the cross-bridge links. The troponin and tropomyosin block more interaction. The muscles relax, but are ready to be stimulated again. So we use ATP for three things. Four things. We operate the pumps, the sodium-potassium pump that repolarizes the sarcolemma, the calcium pump that puts calcium back into the sarcoplasmic reticulum, the energizing the myosin head, putting it into its high energy configuration, and detaching the low energy myosin head from the actin so the whole thing can start again. So there's quite a bit of ATP used. So we have enough ATP made and stored in muscle cells for 5 to 7 seconds of this happening. Now, when we're at rest and not using up the ATP, we make it until the stores are full. And then we transfer those phosphates to something called creatine and make creatine phosphate. And so we can store more energy in the creatine phosphate. When a creatine phosphate, so a high energy creatine with a phosphate attached, meets an ADP, it gives up its phosphate to become the low energy creatine and restore the ADP into an ATP. There's about another 7 seconds worth of energy there. The rest of the energy beyond that kind of 14 seconds, 10 to 14 second mark, has to be newly formed ATP. And this is going to rely on cellular respiration, on Krebs cycle, etc., aerobically and anaerobically. So when there's lots of ATP, then the ATP gives its P to creatine to become creatine phosphate, CP. When ATP levels are low, creatine phosphate gives its P to ADP to make ATP. So the direction of these arrows depend on the concentration of ATP. When it's high, it goes towards the creation of creatine phosphate. When it's low, it goes towards the creation of ATP. So because we've already done this, glycolysis, we get 2 ATPs, but we end up with lactic acid. If there's no air, it's anaerobic, and this is as far as we get. If it's aerobic, we end up with far more ATP. So what we need is oxygen. Skeletal muscle stores oxygen on a pigment called myoglobin. Myoglobin is the exact same as hemoglobin, except that it's found in the muscle. So we can store a fair amount of oxygen this way. The more myoglobin that's in a muscle, the more oxygen that can be stored, and the longer it can go aerobically. Muscles that have endurance tend to be very red. Muscles that create lots of power for a short period of time are less red. Think of dark meat and white meat on a turkey. Turkeys don't fly very far. They don't need any endurance. They need lots of force to get airborne. So their breast meat has very little myoglobin. But turkeys are heavy birds and they walk all day long. Their leg meat has a lot of myoglobin, hence it's dark meat. We've already looked at this, but we get quite a bit of ATP if we can go through the entire aerobic respiration. If we can't supply enough oxygen, then we start to do it anaerobically, and we end up with a byproduct of lactic acid. What we're really looking at is not supplying enough oxygen to fully oxidize the glucose. So when you're finished exercising, you have a buildup of lactic acid, which represents the amount of oxygen that you didn't bring in. So we call this oxygen debt. And that's why you pant at the end of an anaerobic activity. You try and bring more oxygen back in so that you can fully oxidize the remaining lactic acid and make ATP and rebuild up your ATP stores that way. Oxygen debt. Right? That's why we're panting, excess post-exercise oxygen. It may take quite some time to do it, and the more in shape you are, the more rapidly you do it. You repay your oxygen debt. So if we're looking for just going for a walk, low to moderate intensity, we have enough blood flow to supply oxygen for the requirements. If it's really high intensity, bursts, we don't have enough oxygen supply, and so we will use glycolysis for it. So for instance, if you're running the 100 meter, you're running that whole 100 meters, and if you're Usain Bolt in ridiculously short amounts of time. Most track and field athletes are someplace around that 10-second mark. You have enough ATP stored, ATP and CP. To run the 200, you don't have enough time to bring oxygen and make enough ATP. So you end up doing that through glycolysis. The waste product is lactic acid. If it's low to moderate, you end up with aerobic respiration, a lot more ATP, and carbon dioxide. This isn't very efficient, and we end up with a lot of waste heat. A lot of the energy becomes heat. Now, we use this to maintain body temperature, but that's why you warm up when you exercise. When the muscle loses its ability to contract, it is fatigue. Fatigue doesn't mean tired. It means a physiological inability to continue to contract. There's a whole bunch of reasons why it happens, not the least of which is the buildup of lactic acid changes the pH, which inhibits muscle contraction. Motor neurons will run out of acetylcholine, etc., etc. I won't get into what cramping and things are. We will end this video here and continue with the next one.